Thank you so much for joining us today. My name is Kristen Schaefer, and I'm the director at CHE, the Collaborative for Health and Environment. I'm going to get us started today with just some quick housekeeping items. This webinar is hosted by the EDC Strategies Partnership. The partnership members include Healthy Environment and Endocrine Disruptor Strategies, or HEEDS, which is a program of environmental health sciences, Commonweal's Biomonitoring Resource Center, the Health and Environment Alliance, or HEAL, and CHE. We'll disable the chat during the presentation today, but we invite you to type your questions in the Q&A box at any point. We'll get to as many comments and questions as we can after the presentation. If you'd like to activate closed captions, you can do that by clicking the button at the bottom of your screen. This webinar is being recorded. It's scheduled to last for 45 minutes. If you'd like to download today's slides and find links to many of the studies mentioned, visit the webinar page on our website at healthandenvironment.org. We'll put that direct link into the chat now. We're very excited about today's conversation. I'll now hand things off to our moderator, Charles Patton, Director of Commonweal's Biomonitoring Resource Center. Charles.
Thank you very much, and it's with great pleasure that I introduce to you Professor Anne-Louise Ponsonby. She's the division head for the Early Brain Science Department and head of the Neuroepidemiology Research Group at the Florey Institute of Neuroscience and Mental Health in Melbourne, Australia. She's got extensive experience in the design, conduct, and analysis of population-based studies and public health translation. Her work has been on combining population epidemiologic approaches with system biology. She's using this approach to investigate early brain development. In particular, a current focus of her work now is to use this comprehensive approach to better understand the possible adverse impact of some modern chemicals on brain development in early life. Today, she's going to present findings from a study conducted in the Barwon Infant Study birth cohort. This research found that higher prenatal bisphenol A levels are associated with increased male autism spectrum disorders. And as we know, autism in kids is on the rise. I'm not going to go into details, of course, about this study. I'll leave this to her. We should be talking very quickly. But I wanted to mention that she has 427 publications and has contributed to 3 patents. She's on the research committee for the International Pediatric Multiple Sclerosis Study Group and is part of several international collaborations. So welcome, Anne-Louise Ponsonby. Very glad that you're here and pleased to learn more about your recent study.
Thank you, Charles. Pleased to be here. Okay, so the session today is going to center on a paper we published in Nature Communications in August of this year. Male autism spectrum disorder is linked to brain aromatase disruption by prenatal bisphenol A. But I'll also give you a little bit of context for this paper and go into some other issues similarly. So, firstly, many people in this session are here because they're concerned about the growing problem of chemical toxicants, of which plastics are just one. But plastics are the most utilised material in everyday life. And in 2019, we're up to using 460 million metric tonnes. And a key feature is where we are now. The solid line here is the increase in global plastic production from 1950 to 2024. Unfortunately, it's expected to triple up to 2060 unless the situation alters. And at the moment, regulation of chemicals generally assumes safety until proven otherwise. Now, I'd like to point out that with the plastic life cycle, we hear a lot about use of plastics, but there's also a lot of health concerns about the generation of plastics, both for the occupational workers and leakage into the environment, and also in the disposal and waste of plastics and synthetic materials, some of which occurs in the third world. So, sorry, next. So over the time this increase in plastic, there's also been an increase in autism prevalence. particularly since the year 2000. And this is the US Centers for Disease Control figures in the Autism and Development Disability Monitoring Network. Now, it's argued that such an increase might reflect partly changes in diagnostic practice. It's too rapid to reflect genetics. So there's a lot of concern about environmental drivers. And there are many candidate environmental drivers, but, plastics is certainly one of them. For example, the Coke and Pepsi plastic bottles started up in 1978. But of course, correlation is not causation. So we need a lot of good and fine evidence to understand this situation. So before I continue, the rest of the talk is just on plastics. I thought I'd put plastics in the context of manufactured chemicals. So when we usually say plastics, we're usually referring to two things. Plastic-associated chemicals, such as bisphenols and phthalates, that are added to plastics to make them more soft and malleable, or plastics themselves, polymers. And lately, we're particularly hearing about micro and nano plastics. But we need to know that there's also already a lot of adverse information about plastic-associated chemicals, one of which I'll show you today. And then another manufactured chemical that's of great interest is the forever chemicals, such as PFAS, which some of them have half-lives of over 5 years. So they're very long term. And that's not the subject of today's talk. But then, of course, we have other toxicants like pesticides, heavy metals, chlorinated biphenols, tobacco smoke, and so on. And we're not exposed to one at a time. There is a mixture coming in to most of us through ingestion, inhalation, and absorption through the skin. So I just want to put that in context before I focus on just one of these, the bisphenols, bisphenol A in particular. So just this month, we've had a paper published in Nature Medicine on how do plastics, including microplastics and plastic-associated chemicals, affect human health. And if anyone's interested, DOI numbers there that people can access. So they enter the body. They're detected in human samples, including the placenta, and they're associated with adverse health impacts. And the area I work in particularly is neurodevelopmental changes, particularly those that are evident or starting in very early life. And we need to develop and implement global measures to protect public and planetary health. And this Nature Medicine article goes into some of that and is available on the website here. So the plastics have the polymer, then they have the chemical additive. Now, there's more than 6,000 of those, and they have a lot of non-intentionally added substances. One of the key features is that the chemicals aren't covalently bound to the polymer plastic, so they can leach out. It's not like a diamond where you have absolute sort of tying together of all the elements. It's more like a graphite pencil where things are loose and can come out. And 66% of chemicals do not have available hazard data. And of those with data, about 75% are known to be hazardous. And there's limited data on mixtures, a point I'll come back to before. And although there's difficulty in accurately measuring chemicals, there's still a lot that we do know and that we can act on. So now we'll come down to bisphenol A in particular. It's a monomer in plastics. It's even currently in can linings, thermal receipts, food, plastic packaging, and so on. When you see things, it crosses the placenta and the blood-brain barrier, and it's not well regulated, even though it's banned in some products. Other analogues have come in instead that are very similar in structure, like BPS, BPAF, BPF. And we're starting to see in the research that they're causing very similar effects in the lab studies. We haven't really worked them all through into mature human cohorts yet. But at the current state of play before this paper's over 60% of studies, some of which are cross-sectional, show relationship between bisphenols and adverse neurodevelopment. That's not enough to change regulation because 40% don't show that association. Here's an example, a well-conducted review 2021, looking at bisphenol A and phthalates. And it's concluding that prenatal exposure to bisphenol A and phthalates may contribute to neurobehavioral outcomes in children. So with this study that we did, we tried to look a lot more at molecular mechanisms and try and understand the pathway, because we thought that would be helpful in terms of building causal evidence. So you've seen the trends on the left-hand side. But I just want to point out that we think that ASD is complex, it's multifactorial, and both genes and environment are important. But males are in the excess in diagnosis, in some studies up to 80% excess. And one of the things we know is that aromatase, which is a genetic candidate, it's 30% lower in the frontal area of people, males with ASD at post-mortem. That's quite a marked change. And we know that enzyme converts testosterone to neuroestrogen, has a very important role for the male fetus during brain development. So how do we build causal evidence? Pollutants are often not suitable for RCTs. Avoidance trials have been unsuccessful, partly because it's so difficult to eliminate plastics from everyday life, although there are some current trials in progress. At Florey, we've moved to lab experimental studies in tandem with human cohorts. And so these highly dimensioned prospect cohorts take decades to build, and they should remember not to look at one factor at a time, but look at the multifactorial causes of development and exclude confounding and look at other explanations, things that might be associated, like poor nutrition. And they need to be informative enough to give us information on not just the association, but how strong it is, how consistent, how specific, the time relation, and other features. So a key technique we now have available in epidemiology, and I published on this method and others in International Journal of Epi in 2021, was being able to incorporate molecular findings to enhance the quest to better understand multifactorial disease causation. So, sorry, that slides. So this key technique we have here is called molecular mediation, where we model how the plastics influence the molecular pathways. Then we model how the pathways predict different outcomes, such as autism. And we check the proportion of the exposure acting through the pathway to lead to the outcome. And so now we're not just got the association between the exposure of interest and the outcome, a bit like, Joel did with smoking and lung cancer, but we've actually got some molecular pathways we're looking at the same time. So our hypothesis in this study was that BPA in exposure reduces the expression or the activity of aromatase affecting brain development. And because aromatase is particularly important in male brain development, they have a lot more testosterone to break down, and we know males have more activity than females with aromatase, particularly mid-trimester, we expect a stronger impact in males. And then once a molecular pathway is identified, hopefully we can look at a candidate to reverse it, because that would be adding to the causal argument as well, that we really have identified a relevant pathway. So we tested across a multimodal program of laboratory and human prospective cohort studies. And obviously I can't go through in detail in this session, but it included 28 lab studies, 18 of them were mechanistic. 3 looked at human outcomes. We had human and mouse participants. We looked at cell cultures, that's a little yellow block there, and in vivo studies, electrical activity and so on as well as looking at human diagnosis and symptoms for autism and mouse behavior. So the preclinical studies, here's just one example of some of the work we did. The cell expression we show here on the left-hand side, that if we're looking at the aromatase activity, a nice normal activity is about there at about 150. As the cells have more and more exposure to BPA, the aromatase activity goes down. You can see that as being represented as the black groups up there, not as much aromatase is being made. And that's just a control there. Then my colleagues, Wachin Boone and Chris Bassi, used two animal models. Firstly, a BPA, they exposed mice, pregnant mice to BPA mid-gestation, subcutaneous, but they checked they got the same results orally, and they did. And then they thought, well, if it's really acting through aromatase, we should get the same effect in aromatase knockout mice. Now, these are mice that have been bred genetically not to have any aromatase enzyme at all. So they cannot convert testosterone to estrogen. So we were interested to see whether we would get similar patterns across these two groups. And indeed we did. And just to summarize some of the preclinical findings, in the BPA exposed or the ARCO, the knockout aromatase mouse, we got decreased aromatase, brain aromatase expression or activity. And that matches in humans, decreased expression in the ASD brains, particularly in males. And the work we did, which I'll show you in a moment, on epigenetics associated with silencing of aromatase. The animal studies, both the models had an amygdala with decreased dendrite length and neuron number, and that's been previously documented in humans with ASD. They had hypo-responsiveness to social stimuli using a fluorescent marker during viral life called CFOS and an EEG activity, similar to what's been known in humans with functional MRI. Within the cortical layers on the outer brain, there are excess neurons in layers 4 or 5, and those very layers have had excess neurons found at post-mortem in autism, slightly different area though. And the EEG disruption for the BPA or exposed ARCO mouse is similar to the EEG disruption at a similar hertz for humans. And then lastly, we had the social behavior. For the mice, we looked at the behavior in various types of social tests. But for the humans, in the males, we had particularly increased ASD problems or symptoms by age 2 years. I'll show you more in a moment, or diagnosis. And that was using a DSM-5 criteria, so quite a strict criteria in males with low aromatase. So we have some consistency there across a range of findings. So, but quite a useful thing that came out of the work is that Wachin Boone and her team then looked at a whole lot of candidate substances and found one, which she's calling M08 for the moment, that actually reversed the damage caused by the BPA. So, we showed in another set of experiments, it was working through exactly the same aromatase, the same pathways. It was actually, and it was also acting a little bit downstream to sort of compensate for a lack of aromatase. But the encouraging thing was that when we gave this substance to the mice postnatally, we found that they then did not, the BPA mice, who, if they had the M08, they didn't get the problem of the autism-like symptoms and during this social behavior experiment. Now, this is only an experiment in mice, so it's far from, it's on its very early stages, but it has now gone for toxicology study design that's gone to the US FDA for the first stages of being a disease-modifying therapy for people with autism. Because most of the other therapies, of course, they target symptoms like children who can't sleep or children who are irritable. They're not actually targeting the disease process. So now we come to the human cohort studies. So originally, when we were doing our work, we didn't find big patterns for bisphenol A and autism. But after our lab team had been doing all this work, they said, well, you know, you wouldn't expect to find it overall because it's not going to be so important for women. And if people have a very high ability to produce aromatase genetically, perhaps they're not as affected from it. So why don't you go and look in the subgroup of people, this is like personalized medicine approach, that are males that genetically already have pretty sluggish aromatase activity, the low aromatase group. And we built because we had genome-wide association data available in our cohort, we built a genetic score indicating low aromatase activity. And what we found was that for that group, no matter if we looked at age 2, which is red, or age 9, which is the diagnosis of ASD, as opposed to the problems at 2, they did have a higher odds ratio for ASD if they were exposed to high BPA versus low. And you see the bars there exclude this line of 1, which is a 1 times difference, a null effect line. But we didn't see the issue for boys who genetically were able to make lots of aromatase anyhow. And we didn't see this for females, regardless of their aromatase level. Next, we went to a range of epigenetic studies. So with epigenetics, here we have the aromatase gene, and here this little red line shows a promoter of it. And when the BPA comes in and combines with the promoter, it adds methyl groups, and that has an effect in many other studies already been shown of silencing the aromatase gene so it doesn't make as much of the enzyme. So what we found was that women who had higher BPA at 36 weeks gestation, in the cord blood for those children, the aromatase gene was more highly methylated, consistent with being more silenced. And we also found that led to a flow-on effect of having more methylation of brain-derived neurotrophic factor promoter, which itself has been a molecular marker of autism. Now you see there's results of 2 cohorts here because we did this first and then the Nature Communications reviewers said, oh, can't you replicate it in another cohort? And we went, oh my goodness, okay, we'll try. We're fortunate enough to find the Columbia Children and Environment study, which had already published in PNAS that higher BPA was associated with reduced BPA, brain-derived neurotrophic factor. So we said to them, could you come and just look at those results again and see if you've got aromatase as being the middle step here? And they did. And yes, they did have exactly the same pattern. So that was the epigenetic findings. And of course, once you've got low aromatase, you've got lower ability to convert testosterone to estrogen. So here overall is a summary of the findings that are outlined in the paper. And the paper is freely available to anyone who would like to read it. And I was just saying before this started, this took us 12 years to assemble all of these studies. And we think that they're triangulating evidence in that they're all got different approaches and showing slightly different things in different ways. But in particular, we do now think having this reversal of a molecular pathway as well is an interesting thing. Now, for those of you who are involved in policy, I just wanted to highlight a feature of this work, but of lots of work, and that is that we already know aromatase is probably a common site for multiple agents. So there is a need to examine mixture effects. So it's already been published that BPA can silence or shut aromatase, and that's what this study was about. But other studies have shown that phthalates, particularly DHP, can be associated with silencing the aromatase, and so can low vitamin D deficiency. And these haven't all been systematically looked at yet. But, you know, a very important study that we've had published, some of you may know, was by the Endpoint Consortium in the EU in Science 2022. It was called From Molecules to Cohorts. And they found a larger effect on child language delay when they looked at 27 chemicals as a mixture. And they took the mixture, the endpoints for those chemicals from lab studies, and then use those cutoffs to put the 27 together to look at early language development in Sweden, and found that over half the children in Sweden, which is very well regulated, had excessive exposure to the mixture. So the mixture is something we should be thinking of, as well as the individual factors. So our conclusion is that BPA reduces aromatase expression. A live animal study showed us that particularly in the inner part of the amygdala, the part of the brain that not only controls flight, fright, and fight, but is very involved in social behavior. Male BPA exposed and ARCO mice demonstrated ASD-like behavior and brain changes at the structural and functional levels. We also examined female mice for most of these categories and did not find similar consequences. So the BPA silenced the aromatase similarly in both sexes, but the consequences seem to be more adverse for males. And we also observed in a prospective cohort study that males with low aromatase had a greater odds of ASD diagnosis at age 9 and ASD symptoms at age 2 when exposed to high levels of BPA. And this risk estimate was quite high for ASD diagnosis, with the more than 6-fold increase in that subgroup. And of course, we have the epigenetic changes. So that's interesting, but that's just one study. And of course, when we're thinking policy, we want to think about more than one study. And so the last few slides, I thought we would turn to translation of findings from papers like this. Obviously, you know, there may be a therapeutic translation with a new product that is a possible disease modifiable agent that, you know, of course, has to have human trials before we will know that or not. But secondly, this will feed into public policy on what are the safe levels of BPA or plastics or chemicals more generally in early life. And I thought I'd bring up a good news story that we can all do with remembering about, because it makes us feel more cheery and resolute as we go about the difficult tasks of trying to change global health problems. And that is the hole in the ozone layer issue. So as you know, the ozone layer protects Earth's stratosphere by absorbing the sun's harmful UVB radiation. And human-made chemicals began depleting the ozone layer. This was found in 1974, particularly the chlorofluorocarbons and things that were in refrigerants and hairspray. There's a photo of a 1980s hairspray and fire suppression systems. In 1985, the ozone hole was reported, and increased UV radiation reaching Earth. So we have had these sort of scenarios before. Then we had the Montreal Protocol in 1987, aimed at phasing out not one, but more than 100 ozone depleting substances. In 2016, it was amended to include hydrofluorocarbons because they have a global warming potential as well. And they had significant global cooperation, 198 parties, 98% reduction in the use of these substances. And we've had a 20% reduction in ozone depletion from 2005 to 2016. So this picture here is showing you that in 79, it was just starting as a tiny red dot. By 89, that we did have lower levels of ozone down here shown by the red, and that continued to increase. but by the late 2000s, it had stabilized, and now it has actually been decreasing. So what can we do similarly with plastics? Well, in December of this year, there is a global plastics treaty that's being organized by the United Nations Environment Program. They've already had 4 intergovernmental negotiating committee meetings. The 5th is scheduled to occur with this global plastic treaty discussion. And at the moment, there are disagreements across parties about certain aspects of the treaty, but all countries have agreed to work on this intersessional work across countries before coming together to discuss the treaty. In addition, there are other groups, and these are only just 2 of them, but Scientists Coalition for Effective Plastics Treaty, International Organization of Healthcare Without Harm, it's particularly focusing on within hospital plastics, groups such as those hosting and being represented here today. So in conclusion, I'd like to thank the large number of organisations and people that have been involved in this work over time, and the organisations and funding bodies that have contributed, and particularly the participants and families in these population-based studies. Thank you.
Thank you very much for that excellent presentation, for the excellent work you are doing. We have a few questions here, so I'm going to run through those, and then I have a few of my own. Since females have less, why would just low testosterone estrogen cause autism? Is there a theory?
Neuroestrogens, which is different than estrogen generally, but neuroestrogens occur in the brain, and they're known to promote brain-derived neurotrophic factor. They actually increase in the acute stages after stroke. They encourage neuroplasticity, and these are locally produced by the aromatase enzyme from testosterone in males. So in females, some of this process is happening normally, and there are some estrogens coming up from the ovary, but there's also not an override of testosterone. Males are balanced to expect more testosterone, and that's why in a normal male fetus, they will increase the activity of the aromatase to do this conversion during the second trimester of gestation to help the social development of those parts of the brain.
Okay. The substitutes for bisphenol A is the bisphenol S and bisphenol F. Do these 2 substitutions have the same capacity?
Quite possibly. BPF has been shown to be associated with disruption of aromatase in, I think it might be zebrafish, one of the fish species. Certainly in the Selma Swedish cohort, there were adverse child neurodevelopment effects with BPF. So it's a very good point, Shah. We're not saying BPA is the only worry. It's just the oldest one that we know most about. And we can, we've been measuring it a long time, so we know how to do it well. Yeah, so there's no reassurance that the other bisphenols should be safe. And of course, many of them have the same action, the same chemical properties. That's why they've been substituted in. So, yeah.
In terms of you mentioned phthalates, and of course, there's been some studies that indicate that the aberration of DNA expression from phthalates leads to epigenetic intergenerational transfer. So is anything similar happening like that, the intergenerational transfer with the bisphenol?
Well, that's an area of study at the moment, mainly in the preclinical studies, and at the moment to look at intergenerational transmission. But certainly even within one generation, another study of ours that I shared with you was that we found that higher phthalate levels in pregnancy were associated also with higher autism and ADHD symptoms at age 4, but particularly amongst the top 5th of children. We're not talking rare genetics here. We're talking the top 5th of children with low antioxidant capacity genetically. And I think that's really key, that, these plastics, there may be quite large proportions of the population that are particularly vulnerable, and And while we're trying to work out what best to do, there are issues about should we be thinking about personalised prevention and personalised treatment on some of these issues.
And in terms of that, where are you in that discussion and how to take this information and start preventing exposures or the method you mentioned that closed down the bad effects of causing? Where are you in terms of, what's the next step of stopping this?
Well, I myself I stay very much mainly in knowledge generation. So some of our new work will be working in mixtures, or hopefully we'll be working with bigger cohorts where we'll be able to understand genetic susceptibility more. But from the policy side, the Minderoo Foundation, which is a philanthropic foundation in Australia, has been doing, arranged a lot of work of systematic reviews and umbrella reviews on health and the effect of early life plastics, and actually plastics more generally, adult as well, with, later life as well, disease. And all that material's going into the Global Plastic Treaty and has helped the treaty, because it originally started off going to be looking at planetary plastic treaty without the human health. But, the human health is actually quite a significant proportion, as well as the ocean health. So now it's a wider agenda, and I think that can only be a good thing.
Yes, we're grateful that human health, ecosystem health is included now, and much broader in that treaty. It took a lot of work to get that included. People talk about bisphenol N, they talk about avoiding certain plastics or certain household goods and so on. But the problem really, one of the big problems is that the microplastics are basically conveyors of these chemicals. And microplastics are found anywhere clouds are tested around the world, for example.
Absolutely.
Yeah. So
and in fact, we have found in our study that the parents who tried to avoid plastics in pregnancy, they didn't have lower levels than the people who didn't try and avoid plastics. It's so ubiquitous and difficult. So it's not something that an individual person is going to be able to easily change, which makes it a very important issue for population regulators. Yeah.
Well, I think that's very tragic, because people want to do something and they think they can go out and buy the right kind of container for shampoo or something that's going to make a difference. Maybe a tiny bit, but it doesn't take consideration the toxification of the biosphere completely. So that's in terms of like PFAS and other chemicals. It's a real difficult question. How do we stop?
That's right.
And if we can't stop it, how do we help people avoid the results that are going to happen?
Yeah. Well, personally, our study wasn't on it, but personally, from my work and talking to others, I've come to realize particularly bad triad is the heat, plastic, and fat, because most of these plastic chemicals are lipophilic; they like fat. So that particular triad, as many people know, is particularly it does not seem in the studies to date to be in any way something to have happening together. But yeah, the other issues require a lot more work, and it's not, the exposure area is not so much my area as public health. Is more the consequences of the plastics are more my area.
I'm going to jump in, Dr. Ponsonby, until we get Cheryl back, and see if there are any additional ones that we want to bring forth from the Q&As. Let's see. Can autism, you addressed this some in your presentation, can autism be passed down genetically? Example, if the father has autism, the mother has high exposure to BPA, would autism in the child be much higher?
The short answer is yes, but it's a difficult situation. I think the best analogy is a car crash. Car crashes are multifactorial. Say you could say what happens inside the car is like genetics, hey, the drowsy driver. Sometimes a drowsy driver will have an accident anyhow, but it doesn't hurt to be, it doesn't help to be going over a hairpin bend on a day that has rain and the windscreen wipers aren't working and all the things combined together. So we think that for many multifactorial diseases, not just autism, it's this combination of genes and environment that come into play to tip off what we call a sufficient cause and then the disease happens. So one of the areas I worked in was sudden infant death syndrome. And with that disorder, again, it was like a constellation of, if the sleep position was, very important in a lot of cases. That was like the hairpin bend, and when that was altered to sleeping on the back, the SIDS rate went down, but it didn't totally go down, because there were some babies that for other reasons just stop breathing while they're asleep, and we hadn't changed that component cause. So I think that's this car crash analogy is one of the ways to think about it. And it's, I'm an environmental epidemiologist, but I work in genes now because I understand genes are playing a role as well. It's the whole thing coming together that's the issue. And sometimes genes are playing a role just by making us vulnerable to environment. They're not disease-causing genes. They're just making us super vulnerable to whatever adverse environment that things are out there.
Yeah, interesting. Here's a sort of somewhat provocative question. This question is not meant to suggest a pharmacological intervention, but the pesticide atrazine, in contrast to BPA, stimulates aromatase activity. There are multiple human cohort studies in the US measuring atrazine. Is there any indication of a decrease in autism with higher levels of atrazine?
Right. I don't know offhand, but that's very interesting, and I will write that down and make a note of that. I'll be going and checking that later, because it would be, now these papers published, it would be nice to know how all the different things that can regulate aromatase, how do they relate to the downstream consequences, particularly in males. So thank you for that thought, which I hadn't come across before. Thanks.
No, it's interesting. Of course, there are other impacts of atrazine that we'd want to avoid, but
Yes,
it's an
yes.
interesting thing to explore. Charles, are you back with us? Do you want to take a few more questions? Oh, we can't hear you.
That's one of the great things now about some of the work, like some of our colleagues are doing these, with little brain organoids. You can have not just neurons, but microglia and other brain cells as well. And you can actually run toxic products or rescue products across them and see what's happening at the cellular level. So that's the sort of model where you might be able to unravel some of those adverse versus beneficial effects without, compromising, pregnant women issues, which are obviously very, we don't want to intervene unless we're absolutely sure we know what we're doing and that it's a beneficial thing.
Well, I think we're just about at time, so I'm going to go ahead and wrap us up. Wanted to just, first of all, thank you so much, Dr. Ponsonby, for your important research. It's incredibly rich and thought-provoking and interesting. I wanted to also thank Charles for moderating today's discussion, and thanks to all of our participants for the great comments and questions. So I'm going to wrap up now with just some quick announcements. A video recording of today's discussion will be available on CHE's website soon, and you'll all receive an email with a link to that video. Please join us next Tuesday, October 22nd, for a webinar co-hosted with Agents of Change in Environmental Justice. We'll explore the issue of energy and infrastructure justice with current Agents of Change fellows. We'll drop that registration link in the chat in just a moment. Also, we encourage you to support the work of the EDC Strategies Partnership. You'll find links to each member group on CHE's partnerships page. If you appreciate these webinars, please consider a tax-deductible donation to CHE and/or any of these partners. So with that, we'd like to thank all of you again for joining us today, and thanks again to Dr. Ponsonby and to all of our EDC Strategies partners, and to CHE Science Communications Fellow Haley Cavalier for help behind the scenes. Thanks, everyone.